This video is going to talk about horizontal gene transfer. So as we're going to talk about in the future, bacteria reproduce asexually. And we've mentioned this process before. It is called binary fission. And remember, that's where the bacterial cell copies its DNA, and then the cell elongates until it kind of pinches off into two cells, right? So it's gone from one cell to two. And that's how bacteria reproduce asexually. So there's no actual mating between bacterial cells. So there's no, let me think of how to say this. Bacteria are able to get DNA from other bacterial cells, right? But again, that's not considered sexual reproduction. Okay, this is called horizontal gene transfer. They're able to acquire DNA either from the environment or through different organisms or entities. Okay, so we call it horizontal gene transfer because it's not through typical reproduction, which would be vertical transfer, like from parents to offspring is a vertical gene transfer. So there are three different forms of horizontal gene transfer, ways that bacteria can acquire DNA that is not the same as what they already have, okay, and that DNA originated elsewhere, okay? It is not from a parent. And those three forms of horizontal gene transfer are called conjugation, transformation, and transduction. So let's talk about conjugation first. So with conjugation, this process does involve cell-to-cell contact, okay? And the pilus is going to be the structure that mediates this cell-to-cell contact. So if you look at the top picture, okay, originally these cells start out in separate places, okay? They're not always joined like you see there. So I'm going to kind of add to this picture. So here's going to be one cell, and then here's going to be another. Notice one is said to be the F+ donor cell, and the other is an F- recipient cell. So the F+ donor cell has an F plasmid. Remember, plasmids are extra chromosomal pieces of DNA that are not essential for the organism's survival, but it offers an advantage. And in this case, the F plasmid, the advantage that it offers is this organism can form a pilus, okay? And so you'll notice that the donor cell has the plasmid, the recipient cell originally does not, okay? So what happens is this F+ donor cell, who's going to donate part of its DNA, it is going to use that F plasmid to make a pilus, and that pilus elongates. I think of this as like fishing, like when you're casting your line, right? So it reaches out, and the pilus attaches to a neighboring cell, always F-, it does not have the plasmid. And then it's like fishing where it kind of reels in that F plasmid. So I'm going to draw it real small, but essentially that F+ is going to bring that F- cell closer and closer to it, okay, until it's touching like this top animated, not animated, this top illustration here, okay? So it's kind of like the pilus acts like the hook. Once it grabs hold of that recipient cell, then the pilus gets shorter. It retracts it. It's drawing in that recipient cell, okay, until they're close to each other. Now, once that recipient cell is right next to the donor cell, notice how that pilus originally, I'm going to circle it, originally it's like solid, right? Notice that it then gets converted into what's called a mating bridge. Mating bridge. Okay, and so now it is no longer solid, it is hollow. And look at what's happening to this double-stranded DNA plasmid, okay? Notice that one of the strands of the plasmid is going through that mating bridge. Okay, so eventually what's going to happen is there's going to be a single strand of the plasmid in the donor cell, and there will be a single strand of the plasmid in the recipient cell. And then each of those cells can do DNA replication, right? So that at the end of the day, it's horrible drawing, at the end of the day, they both have intact double-stranded DNA plasmids. And so now look, both of them at the end are F plus because the donor cell gave the recipient part of its one strand of its plasmid. After DNA replication, they both have this plasmid. So now both cells can go out and create a pilus and do horizontal gene transfer to a F negative recipient cell. Okay, so some keywords to remember with conjugation are pilus, F plus, also F minus, and cell-to-cell contact. Okay, now with transformation, the bacterial cell is getting DNA from the environment. And this image that I got online is a little misleading. A lot of times this DNA is actually in pieces because it comes from a dead bacterial cell. Okay, remember, a lot of times when bacteria die, right, they lyse, okay, they become pieces. And so when a bacterial cell dies, for instance, right, the DNA also becomes degraded and, is lysed. And so there's pieces of DNA all in the environment that a neighboring cell can pick up. Okay. Now, only certain types of cells can do this that are able to pick up large pieces or large chunks of DNA and let it cross their plasma membrane. And these are called competent cells. Okay, that's kind of a key word to associate with transformation, as is the word environment, because notice there's no cell-to-cell contact here. Okay, so what happens is some of these cells, they're just special in that they are able to take in these large pieces of DNA and allow it to cross their plasma membrane. And then you'll notice what happens is the bacterial cell can normally integrate that piece of DNA into its chromosome. So whatever that DNA coded for, that new protein, that new cell is able to make. Okay, so I don't know, maybe this happened to code for like antibiotic resistance or something. Okay, maybe the cell got lucky and this piece of DNA that it took in codes for an enzyme that gives it antibiotic resistance. Well, now when this cell undergoes DNA replication and transcription and translation, it now gets that enzyme that makes it resistant to penicillin, maybe. Okay. So this is a pretty snazzy process. But in order for this DNA to be able to get inserted into the host genome, it's got to be similar in sequence. Okay, so a lot of times, like this dead organism, maybe it was E. coli. It came from E. coli, cell's dead. Underline it like I'm supposed to. Okay, so then normally it's only going to be taken up by a similar type of organism. Okay, and so because the original DNA came from an E. coli bacteria, if an E. coli bacteria is what takes up that DNA is compatible, right? It's similar. And so it's able to integrate it. Okay, so like oftentimes, like if it was E. coli DNA, but this intact cell is Shigella, there might be too much variation in the E. coli's DNA and the Salmonella's DNA that this type of integration may not be able to occur. But sometimes it can. Okay, so there's a lot that goes into each of these types of horizontal gene transfer, but what we cover at this point in the class is just kind of an overview of what's happening. Okay, so if you're asking yourself, what makes it so competent? There's probably 100 questions that you could ask based off this alone. Just know for right now, the intent is to introduce you to these. And then as we come back to this later, you'll get a little bit more information. Okay, we're not trying to do like information overload right off the bat. Goodness knows you already have enough of that on this exam. Okay, so again, big idea here. There's DNA in the environment from a deceased bacterial cell. Certain types of organisms ones that we say are competent, are able to take in some of this DNA and incorporate it into their genome. Okay, for now, that's all you really need to know about transformation. But I guess a little bit more is the experiment that led us to know that horizontal gene transfer was even a thing, okay? And so there was a scientist named Griffith, and Griffith was studying an organism, I believe it was Streptococcus pyogenes. And you don't need to remember that detail, but that's the organism that causes strep throat, scarlet fever, a lot of other things. It's a pretty dangerous one. And what it found is that one version, one strain of S. pyogenes, when infected in a mouse, it would kill it. Okay, so it's called the S strain. Okay, so this one was pathogenic. I'm just going to write path here. It's pathogenic, meaning it could kill the organism, but they could genetically manipulate this organism. Well, and just because they were mutants out in the world naturally. But when the Streptococcus pyogenes was mutated such that it didn't have a capsule, right, that sugary glycocalyx, without the capsule, this strain of Streptococcus pyogenes was not pathogenic. See, that mouse is alive and well, okay? So this one is not pathogenic because it doesn't have that capsule. All right, so again, they found with the capsule, the S strain was pathogenic and killed the target. Without the capsule, the R strain was not pathogenic, didn't kill the organism. So what Griffith then did was he took this pathogenic S strain, he subjected it to heat. What is severe heat going to do to an organism? It's going to kill it. So this heat killed a strain. When he injected it into a mouse, it survived, right? The pathogen was dead. So there was no harm to the animal. But what surprised and shook the entire scientific community was Griffith then took this heat-killed pathogenic strain, right? So the pathogen was dead, mixed it with living non-pathogenic R strain. And what did you think was going to happen? Well, in theory, the mouse was going to be fine, right? The pathogenic strain was dead. The non-pathogenic strain isn't dangerous. So the mouse should be okay. But it wasn't. The mouse died. And so this provided the first evidence that the non-pathogenic, not dangerous R strain was able to take up DNA from that dead pathogenic strain and become dangerous and become pathogenic, okay? And so when he then infected the mouse, when he isolated the bacteria from the dead mouse, he found that there was both live S and R strains, because those R strains had picked up the DNA, replicated it, transcribed it, translated it, and bam, it had a capsule and it was dangerous, okay? So Griffith's experiment let us know about the process of transformation, which allowed us to know that horizontal gene transfer is a mechanism that bacteria can use to acquire DNA from a different organism. All right, last but not least is transduction. Keyword here is bacteriophage. Okay, remember, bacteriophage only infect bacteria. And so you're already familiar with the lytic and lysogenic life cycles. Okay, so that's really what's going on here, is more like the lysogenic phase. So you'll notice that the bacteriophage is up here at the top. It injects its DNA into the host cell. And then that viral DNA gets incorporated into the host cell. Remember, this is called a prophage. And then remember, this cell is now going to undergo binary fission, right? And so each of these cells is now going to have the bacteria DNA, but it's also going to have that viral DNA. And as the cell goes about its business, I don't have pink, so we're going to go back to red. As that cell is going about its business, living its best life, right? It's going to be replicating, transcribing, and translating whatever is on that viral DNA. And so sometimes those viruses come with a toxin. That's how they make things sick. And so now this bacteria has that gene for the toxin as part of its own genome. And so as the bacteria undergoes transcription and translation, it's creating that toxin. So if that bacteria infects a host like us, it now has this extra weapon, this toxin that it can release into the host and give that give that bacteria an advantage, right? The toxin is going to make the host sick. That's what the bacteria wants, right? Because it can then multiply and stuff within that host. So when the bacteria has acquired DNA from a virus, that is called transduction. Okay, it's always via bacteriophage, and then another keyword there is prophage. Okay, so this is how the bacteria has acquired DNA from a virus. All right, y'all, that's it. Let me know if you have any questions.